Yes, from Bryophytes today we are going to discuss Anthoceros Polytrichum in detail along with the ecological and economic importance of Bryophytes. So, first if you see the schematic position of Anthoceros, Anthoceros belongs to the division Bryophyta, class Anthoceratopsida, order Anthocerataceae, family Anthocerataceae and genus Anthoceros. Okay, this is the systematic position of Anthoceros. Look into the distribution and habitat. It contains around 200 species and it is widely distributed, present everywhere. It is cosmopolitan in its distribution. Okay, it is cosmopolitan in its distribution and it has around 200 species. It grows in moist, shady habitats like slopes and rocks and ditches and over decaying wood as well. Poorly, these are adapted to some dry conditions. These are adapted to dry conditions. In India, around 25 species are recorded. Okay, around 25 species are recorded in India. The common Indian species, if you see, Anthoceros himalayensis, Anthoceros erectus, Anthoceros chambensis. These are the common Indian species of Anthoceros. Common Indian species of Anthoceros. These are found in western Himalayas on an altitude ranging 5,000 to 8,000 feet and in regions like Mussoorie, Kulu, Manali, Punjab, Chamba. In these regions you can see, in high altitude regions you can see wide range of Anthoceros. Now look into the morphological structure. Anthoceros has both dorsal and ventral surfaces. It is a thalloid structure. Gametophyte is thalloid. It is dorsiventral, has both dorsal end and ventral surfaces. Okay. So, this is dark green prostrate in habitat. It shows dichotomous branching. Dichotomous branching is observed, same like that of Mercantia, like how you see dorsiventral in arrangement and dichotomous branching. Fine. This forms orbicular or suborbicular rosette like structure. So, if you observe in ventral surface there are mucilaginous cavities. These mucilaginous cavities will allow the symbiotic association with blue-green algae, for example, nostoc. Okay. This will allow symbiotic association of blue-green algae, for example, nostoc. And stomata like slits are on the dorsal surfaces. Stomata like slits. Are present on dorsal surfaces. You can see it, right? Unicellular smooth walled rhizoids, they are present on ventral surfaces. Mucilage cavities are present on the ventral surface. This contains blue-green algae like nostoc, which are in symbiotic association, which will help for nitrogen fixation. Stomata like slits, they are present on the dorsal surface. Here if you see, thallus surface variations are present. There are many thallus surface variations. You can see smooth surfaces are present in Anthoceros laevis, velvety type of surface, thallus surface is observed in Anthoceros crispulus and rough or spiny type of surface is observed in Anthoceros fusiformis. Midrib is totally absent and thallus is thicker in the middle. In middle region, the thallus will be thicker. Okay. Now, you will observe both ventral surface features and dorsal surface features. Ventral surface will be having rhizoids, rhizoid types if you observe tuberculate rhizoids with scales are present and it mucilaginous hairs are absent. Mucilaginous hairs are absent, but it has tuberculated rhizoids with scales. Mucilaginous cavities are as dark blue-green spots present in the nostoc colonies itself. In the dorsal surface, you can see stomata like slits which secrete mucilage, but on the ventral surface you can see symbiotic associations of algae. These mucilaginous cavities are observed in what color? Dark blue-green spot color. So, remember the morphology of Anthoceros is dorsiventral, dichotomously branched, and on the dorsal surface you have stomata like slits which will secrete mucilage. On the ventral surface you have symbiotic associations of blue-green algae, that is nostoc, which is present in the form of dark blue-green spots. And One more thing to be highlighted here is about rhizoids which is present on the ventral side. What type of rhizoids are present? Tuberculated rhizoids with scales. This is important from this slide about morphology of Anthoceros. Now you have understood the gametophytic structure. You should know clearly on the sporophytic structure and how sporophyte is emerged on the gametophyte. Look into here. Sporophyte is observed during September to October month region, and these are erect cylindrical branches. If you see, each sporangium is surrounded at the base, it has sheath like involucre. At the base it has sheath like involucre. Each sporangium at the base will have sheath like involucre. Internal anatomy of Anthoceros thallus. Internal anatomy of Anthoceros thallus. There is no tissue differentiation, uniformly composed parenchymatous cells are present. Okay. No tissue differentiation, but it has uniformly composed parenchymatous cells. And here you see thallus thickness. Thallus thickness. Anthoceros leaves, punctatus and crispulus. No need to remember like how many cells are present in each species. Just remember the midrib present in the thallus will be thick, will give overall range of thickness for the thallus in the Anthoceros. And if you observe the epidermis, two epidermal surfaces are present now. You can see upper epidermal surface and lower epidermal surface. If you observe upper epidermis, it has large lens shaped chloroplast. Chloroplasts are present on the upper epidermis. On the lower epidermis, it has tuberculated rhizoids with scales. And each cell in the epidermis, it has large discoid oval shaped chloroplast. Chloroplasts will contain pyrenoids. This is the similar feature of algae. In the beginning also we have learnt. Similar feature of green algae is having chloroplast. Inside chloroplast, the storage bodies called pyrenoids are also present. Each pyrenoid is an aggregation of 25 to 300 spindle shaped bodies, remember. And then about cavities and pores which are present. You can there are no air chambers or air pores, but mucilaginous cavities are present on the ventral side. Okay, there are no cavities or pores, but mucilaginous cavities are present along with symbiotic association. They are appeared as dark green spots. So here you observe first they are formed schizogenously, means by cell breakdown in the epidermal region. When epidermal cells break and when some space is created in between, at that region mucilaginous cavities have emerged in the ventral surface of the epidermal region. They open via slime pores with non-functional guard cells. Okay. They have slime pores and non-functional guard cells over the surface. And nostoc colonies cavities, they open via slime pores. What is the ecological significance of nostoc symbiosis, of symbiotic association with nostoc? What is the ecological significance? Here we go. Nostoc, there is a mutual benefit with it. Anthoceros will provide carbohydrates to nostoc, and nostoc will fix atmospheric nitrogen. Fine, right? Anthoceros will provide carbohydrates and nostoc will fix the atmospheric nitrogen. This is the significance of symbiotic association. Now look at this table carefully. What are different modes of vegetative reproduction in Anthoceros? In the species, in which type of species, what type of vegetative reproduction is operated? Look at here carefully. First is fragmentation. Fragmentation is the most common mode of vegetative reproduction. Second is by tubers. Okay, by tubers, thickened marginal thallus tissues, they form perinating tubers under stressful conditions. They form perinating tubers. If you can see, Anthoceros hali, personi, thallus margins will be formed, thick thallus margins will be formed. From there tubers will be developed and their dissociation falling onto the suitable substratum will give rise to a new thalloid body. Next is gyma. By gyma is the most common mode of vegetative reproduction in Marchantia, but you can also see this in few species of Anthoceros. Very important. Remember those 3 species ma, like Anthoceros glandulosus, Anthoceros propaguliferus and Anthoceros formosae. These are the 3 species of Anthoceros in which gyma will be the reproductive, gyma will perform the vegetative reproduction, wherein single gyma will give rise to 2 new thalli. Single gyma give rise to 2 new thalli, and persistent growing apices, the apical regions which will survive the unfavorable conditions, the apical regions which will continue to grow, those apical regions at the apices, they detach from the parent plant, and these apices landing onto the suitable substratum will grow into a new thalli. Next is apospory. Okay. Next will be apospory. Apospory, here sporophytic cells, for example, capsule and intercalary zone form diploid gametophyte like thallus. Okay. So, sporophyte cells, they form diploid gametophyte like thallus. So, apospory type of vegetative reproduction is observed in Anthoceros levis, which is discovered by Lang in the year 1901 and Schwarz and Bach in the year 1926. Do not remember about this is not required. Just remember apospory type of vegetative reproduction is observed in which species of Anthoceros, that is enough. Next. Yeah, sexual reproduction in Anthoceros. We observed that there are 3 modes of sexual reproduction: isogamy, anisogamy and oogamy. Oogamous type of sexual reproduction is most common in Anthoceros, wherein male gametes will be biflagellated antherozoids and female gamete is egg, which is present inside the female reproductive structure, that is archegonium. Okay, inside archegonium, which is female sex organ. Fine. Here inside the archegonium, it has the female gamete, that is egg. So, you observe monoecious species are bisexual. They have both male and female sex organs in the same thallus, observed in Anthoceros longi, golani, fusiformis, punctatus, crispulus and himalayensis. These are the diverse species which have heterothallus, means it has both male and female sex organs on the same thallus. Dioecious or unisexual or homothallic. Examples you can see: Anthoceros erectus, chambensis, Halley, Personi and Levis. These are different species which show dioecious type of sex organs, where both male and female sex organs are on the different thalli. So, here you observe monoecious species are protandrous, which mean male sex organs mature prior to the female sex organs. Male sex organs mature prior to female sex organs. This is a mode to escape autogamy. Fine, clear? I hope you are clear with monoecious, dioecious, examples of Anthoceros and autogamy. Like what is the mode that is being performed to escape this process? Okay, protandrous. How male sex organ is formed? Structural development of antheridium. Observe it carefully. How the antheridium looks like? It is like club or pouch shaped with a stalk. Okay, it is club or pouch shaped with a stalk. How is it developed? Look at here carefully. One of the superficial fertile cell will undergo a transverse division, and after one transverse division, apical roof initial is formed, and at the base you can see antheridial initial, roof initial and antheridial initial. Now after anticlinal divisions, this forms 8-celled structure. Later, after complete anticlinal and periclinal divisions of antheridial initial and roof initial, it will undergo both anticlinal periclinal division, forming out a jacket layer. Roof initial will form what? Jacket layer. Antheridial initial, after undergoing mitotic divisions, this forms a tissue of androgonial chambers which contains androgonial cells. Clear, right? So, one superficial fertile cell undergoing transverse division forms outer roof initial and inner antheridial initial. Inner antheridial initial, after sequential mitotic divisions, this will be forming out what? Androgonial chambers, inside which androgonial cells are present. And outside roof initial, this roof initial after sequential mitotic divisions forms out a complete jacket layer over the antheridial chambers. Clear till here? Any doubt anywhere? So, here antheridium shape is club or pouch shaped and it has a stalk. Stalk is how many rod? 4 rod or massive. It is species dependent. You can see Anthoceros punctatus and Anthoceros erectus. These have slender stalks, and Anthoceros leavis, more massive species are formed. You can see one or more antheridia per antheridial chamber. One antheridial chamber can have many antheridia. Single layer jacket, sterile jacket which will enclose the complete androcytes, means antheridial cells are enclosed by complete jacket layer. So, mature antherozoid and antherozoids which are released from the androcytes, these antherozoids are unicellular, uninucleate, biflagellated, and it has a linear body with biflagellated and uninucleated structures. And what type of color will be there for male sex organs when you observe it under microscope? It is green to red or bright orange because of plastid color shift. Fine, right? So, this is about shape or structure of antheridium and how the antheridium structure is being developed. Now, development of antheridium, just now we have seen. Antheridium is endogenously formed, means inner antheridial cell only is maturing to complete antheridium. Okay, inner antheridial cell which is formed undergoes sequential mitotic division and antheridium is formed. So, antheridium is endogenous in origin. Fine, right? And this and all we have discussed in the previous slide itself. Just go through the process. If you are having any doubt, you just ask me. Fine. Now, dehiscence of antheridium. Okay. Here you look into water aids in dehiscence. Water aids in dehiscence. Okay. Antherozoids will swim across the water. That is the reason we call bryophytes as what? Amphibians of plant kingdom, because they are dependent on water for fertilization. Roof initial cell, it will break, the chambers of antheridia will be braked initially. Roof of antheridium chamber will break and antheridia will absorb water, thereby antherozoids will be released. And how they reach up to the female sex organ? The movement is guided completely by chemotaxis. It is chemotactic movement. Fine, right? The chemicals which are being released out by the female sex organ are being attracted by antherozoids and it will reach the female sex organ, that is archegonium, and it will cross across the neck canal cells, venter canal cells, and it will reach the egg cell. So, if you see a mature archegonium, where is it present and what is its structure? It is marked by mucilage mound externally and it develops within dorsal thallus tissue. And here mature archegonium if you see, cover cells which are covering up the neck canal cells and venter canal cells will be 2 to 4. We have 2 to 4 cover cells, followed by how many neck canal cells? 4 to 6 neck canal cells. Okay, 2 to 4 cover cells and then 4 to 6 neck canal cells, and one venter canal cell at the base. Below the venter canal cell we have one egg cell. So, egg cell is at the last and at the base of the venter canal cell. Jacket not distinct from the vegetative cells. Okay, jacket is not distinct from vegetative cell. So, this is the mode how antherozoids will reach the female sex organ, and this is the structural description of Archegonia in Anthoceros. What is the development of Archegonia? Same here, like how Antheridia, one superficial cell will undergo the divisions, here also the same pattern will happen. One superficial cell which is archegonial initial, they will divide and form, it will divide and form upper archegonial cell and lower primary stalk cell. Okay, upper archegonial cell and lower primary stalk cell will be formed. Archegonial cell will further divide and it forms 3 jacket initials, 1 axial primary cell. Here, how many jacket initials are present and what do they form? Look at here carefully. I will give you a clear flow chart. Yeah. First, one superficial cell undergoes transverse division, forming out outer cell which is primary archegonial cell. And lower primary stalk cell. Fine, lower primary stalk cell and upper will be archegonial cell. So, here primary stalk cell, if you see different species, Anthoceros erectus, there is no division and this functions directly as primary cell. Archegonial cell will divide and here you see 3 jacket initials, archegonial cell will further divide, forming out 3 jacket initials and 1 primary cell, 1 axial primary cell. Fine. And here you see jacket initials, what do they form? 6 rows of neck cells and a venter wall is formed. Axial cell will divide further and outer cover initial and inner neck canal initials are formed. Neck canal initials will give rise to 4 to 6 neck canal cells. Venter cell will give 1 venter canal cell and 1 egg cell is formed. Clear till here? This is the differentiation. This is how the archegonium complete structure is differentiated and formed from a single superficial cell. Okay. I hope the divisions are clear to everyone. And now, fertilization in Anthoceros. As I told you, the archegonium is covered by mucilaginous core and this will release out chemical attractants and this will trigger chemotaxis of antherozoids. Thereby it will reach the egg cell, and after fusion of the nuclei between antherozoid and the egg cell present inside the archegonium. Here you can get one question like, what is the site of fertilization? Site of fertilization is archegonium. Inside archegonium only fertilization takes place. And it forms a diploid zygote which is addressed as oospore. This forms the first cell of sporophytic phase. Fine. Inside archegonium, which is site of fertilization, post fertilization zygote is the first cell formed which is diploid. Zygote is also addressed as oospore, and this is the first cell of sporophytic phase. Fertilization will mark the end of gametophytic phase. Clear till here? Any doubts anywhere? And then this is the complete life cycle, if you can see. Sporophytic phase, zygote development, post fertilization what will happen? Zygote will enlarge, and around the zygote you can see it will secrete a cellulosic wall. First division will be vertical and second division will be transverse. Okay. Second division, which is transverse, it will form a quadrant stage where upper cells will be larger and lower cells will be small. This quadrant stage undergoes one more division and it forms octant stage, which is 8 celled, and 4 cells are arranged in 2 tiers. 2 tiers, 4 cells will be arranged. In different species the arrangement varies. For example, Anthoceros erectus, if you observe, lower tier is foot and upper tier is seta and capsule. Complete 4 cells are formed like this. Okay, this is lower tier and here it is upper tier. Okay, lower tier will be foot and upper tier is seta and capsule in Anthoceros erectus. Anthoceros fusiformis, Pearsoni and Himalayensis, upper tier will again divide into 3 layers. Lowermost foot, middle region will be meristematic zone and upper region of the upper tier will be capsule. Like that the differentiation patterns will vary in different species of Anthoceros. Clear? And next, foot development, how foot and capsule are formed. Seta of course you cannot find, but you can see foot and capsule in Anthoceros. Foot, lower tier will turn to foot. Lower tier of octant embryo will turn to foot. Foot will be of what shape? Bulbous parenchymatous foot is formed at the lower end. In Anthoceros punctatus, outer foot cells they form palisade layer, and in Levis himalayensis, superficial cells they form haustorial projections. Fine. And if you see capsule development, outermost tier, this will divide transversely, outermost tier divides transversely, forming 2 to 3 tiers. After periclinal divisions, this forms outer layer, that is amphithecium, and inner core, that is endothecium. Amphithecium and endothecium. Endothecium will form a sterile columella. Fine, right? This will start 4 cells and matures into 16 rows. 4 cells and then it will mature to 16 rows. Amphithecium will be the outermost jacket. This forms jacket initials and fertile layer. This forms jacket initials and fertile layer. Jacket initial will further undergo division, forming out epidermis, and epidermis contains stomata in their mucilaginous cavities. Inner layers will contain chloroplast. Chloroplast contains storage bodies called as pyrenoids. Clear? This is about the sporophytic development in Anthoceros. Now you see sporogenesis. Inside sporophyte, how spores are formed. Archisporium, which is diploid, this will give rise to spore mother cells. If you can see, spore mother cells are present. These spore mother cells, ploid is diploid, and spore mother cells are again enclosed by a jacket outside. Jacket layer is enclosing the spore mother cells. Here you see spore mother cells, they are oval dense cytoplasm and they have large nucleus. They undergo meiosis. First they will undergo meiosis, giving out a spore tetrad. Spores will be haploid. And how the spores will be dispersed? Spore dispersal is mediated by elater cells. These elater cells are formed from elater mother cells. They divide mitotically, forming 4-celled elaters. Elaters are 1 to 3 celled units. They will break into 1 to 3 celled units called as pseudo-elaters. So, the pseudo-elaters and elater cells, they will help in dispersal of spores. There are no thickenings and bands, unlike true elaters. Okay. So, you just observe that elater mother cells undergo mitotic divisions, whereas spore mother cells inside undergoes meiotic divisions forming out spores. Elater mother cells outside, they undergo mitotic division forming out elaters. If at all few elater cells will break further into 1 to 3 celled units, they form pseudo-elaters. Both elaters and pseudo-elaters, they do not have any thickenings, but they will help in release of the spores. Next is capsule elongation and protection. Capsule elongation and protection. First, meristematic zone, which is the middle tier, which is intercalary. This grows continuously, producing out a capsule tissue outside. On elongation of the capsule, this is surrounded by involucre. Capsule is surrounded by involucre, which is a protective sheath. Okay. And this is derived from partly archegonial tissue and partly gametophytic thallus. In early stages, sporophyte is completely enclosed. Fine? Clear till here? Any doubt anywhere? And then alternation of generations in Anthoceros. Life cycle will show alternation of generations. You see, gametophytic phase, which is haploid, will alter with sporophytic phase. Sporophyte is dependent on gametophyte. And what will be the sporophytic structures? Foot and capsule. Foot and capsule. There is no seta. Okay. And gametophyte structure will bear sex organs, antheredia and archegonia. Whose ploidy is haploid. And here this ploidy will be diploid. Remember the ploidy, it is very important. And then, yeah, this is clear to us. Now, if you observe the life cycle, heteromorphic life cycle is seen, wherein gametophyte and sporophyte, they are morphologically distinct, and diplohaplontic, both haploid and diploid stages are multicellular and they are alternating with each other. Sporogenic meiosis is occurring in sporophyte. Meiotic division, what type of meiosis is being seen? Sporic meiosis, that also takes place inside the sporophyte. Here once you see the complete life cycle of Anthoceros. Complete life cycle of Anthoceros. First, you observe here a mature thallus which is haploid. This has both male and female sex organs. Male sex organ produces biflagellated antherozoid which is haploid, and female sex organ archegonium will have a haploid egg cell. Post fusion, antherozoid and egg cell, post fusion, this forms diploid zygote which will mature to embryo. Embryo will undergo mitotic divisions and it forms young sporangium. Sporangium in the capsule region. In the capsule region of sporangium, this sporangium consists of spore mother cells. Spore mother cells are diploid and they will undergo reductional division or meiotic division. Meiosis will happen. Post meiosis, haploid spore tetrads are released. Spores are being released, and this haploid spores landing onto the suitable substratum will develop into a young thallus which is haploid. So, here you can see equal haplontic and diplontic phases, equal stages of gametophyte and sporophyte, and one thing to be noted, gametophyte is independent whereas sporophyte is dependent on the gametophyte. So, here you can observe, right, haplodiplontic life cycle observed. Next, Polytrichum. Yeah, Polytrichum you can observe, it is in the genus which is Polytrichum, Polytrichaceae family, Polytrichales order, Bryopsida is the class, subdivision is Muci and division is Bryophyta. Occurrence, Polytrichum is observed worldwide. It is common in cool temperature and tropical regions as well. Typically they grow in cool and shady places. Plant body of Polytrichum is usually gametophytic plant body and main plant body is haploid gametophyte. Okay. So, adult plant will consist both rhizome and upright leafy shoot is present. Okay. It consists of rhizome and upright leafy shoot, which is little advanced bryophyte. And in rhizome, if you see TS of rhizome structures, previously you have observed rhizoids, but here rhizome structure will produce rhizoids. Okay. Look into it. This grows horizontally and it has 3 rows. Small brown colorless leaves are present which will produce rhizoids. They arise at the base of erect gametophores and it contains protoplasm and oil globules. This will function as mechanical support to tall gametophores. Upright leafy shoot, if you see, this leafy shoot is conspicuous, grows upwards from rhizome, and they bear large leaves which are spirally arranged. Leaves have broad base, upper leaves are green and lower will be brown. These are further divided into sheathing leaf base and narrow distal limb or leaf blade. Extra photosynthetic tissue which is known as lamellae is present. These lamellae are vertical plates of green cells, remember, most important statement. What are lamellae cells in Polytrichum? They are vertical plates in the green cells. They enhance photosynthesis. Here if you see TS of the structure, you can observe leptoids and hydroids. These are the structures which will conduct water and food materials all across the thallus. Okay, and amylome is the storage tissue which is present at the center. Amylome is the storage tissue which is present at the center. Clear till here? This is the structural description of what rhizome and leaves. What type of leaves are present? 2 types of leaves, scaly leaves and foliage leaves. 2 types of leaf structures are present, scaly leaves and foliage leaves. And here you can see 2 epidermal layers again. You can observe scaly leaves and foliose leaves and 2 epidermal layers. Lower epidermis is present, upper epidermis is also present. And above the lower epidermis you can see a clear mass of parenchymatous tissue is present. Parenchymatous tissue. Above parenchyma, parallel rows of lamellae structures are present. These lamellae structures are green in color and they perform photosynthesis. And these lamellae structures contain chloroplast. Fine. This also possesses lower epidermis, at the above it also possesses wing. Fine. So now you see scaly leaves will be small, brown and colorless. It has rudimentary blade which is spirally arranged on the axis. The leaves are spirally arranged on the axis. Foliage leaves, they are present only on the upper branch portions. Each leaf will have 6 to 10 mm long. They are differentiated into proximal sheath base and distal narrow limb. Clear till here? And then anatomy of Polytrichum. Leaf anatomy just now we have seen. Lower surface it has single layer epidermis. Above the epidermal structure you can see parenchymatous tissue is present. Okay. And above epidermis will be 2 layers. Lower epidermis will be single layered and upper surface or upper epidermis will be sclerenchymatous tissue and it will provide mechanical support to the leaf structure. Central region is completely filled with parenchyma and it has numerous lamellae. Lamellae will conduct photosynthetic activity. Quite a times the questions have been asked in your previous year papers if you see on lamella structure of Polytrichum. Lamella structure of Polytrichum will contain chloroplast and they will conduct the photosynthesis, which these lamella structures are present above the parenchymatous tissue as we have seen in the diagram here. Clear? Next, stem anatomy of Polytrichum. TS of stem will give you out medulla, cortex and epidermis. Epidermis is the outer region and in the middle cortex will be present. Inner medulla is there. Inner medulla is further differentiated into central zone and peripheral zone. Okay. Cortex is thick walled. Cortex is thick walled with cells which support the cortex region, and these strands are called as what? Mantle. Cortex will have many conducting tissues inside it, which is known as mantle. Mantle cells will also contain starch grains. Okay, mantle cells contain starch grains in it. Fine. Epidermis, which is the outermost layer, it has single outer protective layer. Okay, epidermis has single outer protective layer. This is present above the cortex region. Polytrichum life cycle, you see. First, vegetative reproduction, it can be either through protonema, vegetative buds, or by fragmentation. What is protonema? Protonema is the primary multicellular thalloid structure. Okay, when haploid spores lands on the substratum, this forms primary gametophytic structure which is protonema. Protonema undergoes lateral budding which gives rise to leafy gametophytic structure. This gives rise to leafy gametophytic structure. Fine, right? And then sexual reproduction. Dioecious plant, Antheridia and Archegonia, they occur on the separate plants. On two separate plants, this dioecious, this Antheridia and Archegonia are present, male and female sex organs. So, this Polytrichum is what? Dioecious plant. Antheridial head is surrounded by perigonial leaves, and they also contain some supportive structures over this, which are known as paraphyses. Mature Antheridium will have androcyte mother cells. This androcyte mother cells will give out biflagellated sperms, which are antherozoids. Fine. And then development of Antheridium is from single antheridial initial. One single antheridial initial will divide into stalk cell and antheridial mother cell. Stalk cell and antheridial mother cell. This is surrounded by jacketed layer of cells which are single layered wall. This jacket initials form a single layered wall around them, and inner mast cells are androcytes which will contain antherozoids, those are sperm cells. And when these antherozoids are released into the water, the operculum opens and this forms sperm mass is released out. Operculum will open and sperm mass is released out. Clear? And then here you can see the clear formation of complete mature Antheridium. So, one primary cell of Antheridium divides into two, Antheridial mother cell and an apical cell, turns to stalk cell, and here Antheridial cells will further divide, and you can see they are surrounded by the segments or structures called as paraphyses. Inside the Antheridium structure which is formed, you can see androcytes. Androcytes will be releasing out the antherozoid structures. Clear? Next, archegonial head, female sex organ. Look at here, archegonium head. Archegonia are born at the stem tips by pericytial leaves. By pericytial leaves, where are archegonia present? Over the stem tips. And each archegonia is flask shaped with long neck and thick stalk. It has 6 rows of cells. 6 rows of cells which are canal cells. They form canal cells inside. Both venter canal cells and neck canal cells are formed inside. Okay. And venter will have venter canal cells, and at the base of venter canal cells you will have the egg cell. Clear? And then you see development of archegonium. Archegonium development will start from a single cell, that is archegonial initial. This will divide forming stalk cell and body cells. Body cells and stalk cells. Fine. So, central cell will be forming up egg and ventral canal cells. The neck formed, they are from apical divisions, and canal cells will cut off from the apical divisions. Fine, right? Now, so this is how the archegonia is developed. Archegonial initial cell will first divide, forming up the apical cell and the stalk cells, and that will undergo further division, forming out the complete archegonium structure, which is surrounded by the jacket of layers. At the base you can see egg cell, above which ventral canal cells are present, above which neck canal cells are present. This neck canal cells and ventral canal cell will mediate the movement of antherozoids. And how antherozoids are dispersed? Antherozoids are dispersed through water, and they will reach out the egg cell. And here also the site of fertilization is inside archegonium, post which zygote is formed. Zygote will undergo the mitotic division, sequential mitotic divisions, forming out a sporophyte. How the zygote will divide, you see. How the zygote is dividing. Zygote will undergo a transverse division. Zygote undergoes a transverse division, forming an outer epibasal cell and a lower hypobasal cell. Outer epibasal cell is formed and lower hypobasal cell is formed. Here embryo will completely form an amphithecium and endothecium. Embryo forms amphithecium and endothecium. Amphithecium is capsule wall, epidermis and spongy tissue. It forms, amphithecium forms all these structures. By undergoing mitotic divisions, it will form capsule wall, epidermis and spongy tissue. Inner layer is completely formed by endothecium, which consists columella, spore sac and archisporium. Remember, archisporium is the cell which is forming out the spore mother cells. Spore mother cells are diploid, and this spore mother cells will undergo reductional division or meiosis for releasing out the haploid spores. It is very simple. Once again you listen. Post fertilization, what happens? Zygote will divide, will undergo transverse division, forming out outer epibasal cell and inner hypobasal cell. Then it forms a complete embryo. Embryo will have amphithecium and endothecium. Amphithecium will generate out capsule wall, epidermis, spongy layer, whereas endothecium internally forms columella, spore sac and archisporium. Archisporium differentiates into spore mother cells which are diploid, and this spore mother cells undergo reductional division, forming out haploid spores. Clear, right? Now you see mature sporogonium structure. Foot, seta, capsule, three are present. If you see Anthoceros, it just has foot and capsule, but here it has, here in the Polytrichum it has foot, seta and capsule. Foot is absorptive, embedded inside the gametophytic tissue. It will absorb the complete nutrients and it will take a strong structural support to fix over the gametophytic structure. Next is seta. Seta is a long tissue which will support the foot. It will connect the foot and above capsule. Okay. So, here seta is long, supportive with conducting tissues, and capsule which is present at the above, it has apophysis, theca and operculum. Apophysis is the lower part of the sporophyte of the capsule. The lower part of the capsule is apophysis. You can see here lower part of the capsule is apophysis. It will connect to the seta. Okay. And theca, theca is the complete cavity or layer which will bear spore producing region with multi-layered wall. And operculum above, here you can see, right, operculum above is having a conical lid which will support the epiphragm and peristomic teeth. Here if you can see this operculum, it will be having the peristomic teeth, like how elaters and pseudoelaters in Anthoceros will mediate the spore dispersal. In Polytrichum you have peristomic teeth which will ease or aid in spore dispersal mechanism. Till here it is clear, right? And then here we go with spore structure. Once the spore is landed onto the suitable substrata, how the spore is going to germinate. Spores are usually yellow unicellular and it has two thin walls, that is exosporium and endosporium. It has exosporium and endosporium. This germination of exosporium, if you see, exosporium will rupture and here you observe endosporium, it forms the primary gametophyte, that is protonema. Protonema undergoes lateral budding and these buds will develop into a new gametophyte. Fine. And here you can observe alternation of generations. Haploid dominant gametophytic phase will produce both Antheridia and Archegonia which are haploid. Over this haploid gametophyte you can see diploid sporophyte which is formed. Sporophyte is dependent on the gametophyte and you can see clear alternation of generations. Two generations are observed. Clear, right? So, here you observe first gametophytic structure. Yeah, haploid gametophytic structures. It has both male and female sex organs. Both are haploid, okay, and it forms a diploid zygote inside. Zygote will undergo division, forming out a mature sporophyte where spore mother cells are surrounded by a theca. Okay. Spores are dispersed through peristomic teeth. Peristomic teeth which will aid in spore dispersal. These spores landing onto the suitable substratum, this will form a primary gametophytic structure which is protonema. This will mature into a mature gametophytic structure by undergoing lateral budding which will bear male and female sex organs. This Polytrichum species are what? Dioecious or unisexual, means both male and female sex organs are present on two different plant bodies. Fine, right. So, with this we are done with the life cycle of Polytrichum, which is haplodiplontic life cycle, and dominant plant body is gametophyte. Sporophyte is dependent on gametophyte. Clear with Anthoceros and Polytrichum? Any doubts anywhere, ma? Next, economic importance in bryophytes. Yeah, economic importance in bryophytes. The primary most importance is peat formation. Look at here, sphagnum, which is a bryophyte, is also addressed as peat moss, cotton moss, bog moss. Sphagnum is addressed as peat moss, bog moss or cotton moss. First, peat moss, you see, why is it known as peat moss? Because sphagnum is the main component of peat which is used as fuel, source of ethyl alcohol, peat, tar, ammonium and paraffin. This is the major source, and it is a good soil conditioner. That is the reason it is also known as cotton moss. And sphagnum is used for the transshipment of commercially important plants, as this can absorb water. It can be a good conditioner for water, and it is easy for the transshipment of commercially important plants by using sphagnum. So, it is also known as cotton moss. It is also known as bog moss because sphagnum can grow over the wood like structures, long block or wood like structures, sphagnum can start the succession over it. Okay. So, that is the reason it is also known as bog moss. The pioneers of succession are of course lichens. Lichens will start up the succession process, but after lichens it is bryophytes which form a dense like mat over the soil surface, and the major component of the bryophytes, major species is sphagnum only, which will form the dense mat in the succession. After lichens it is bryophytes which are important in the succession process, and it is important for water retention and transportation. Just now we have learned soil formation, that is the reason it is also known as bog moss. Animal bedding, some bryophytes will serve as soft bedding for animals. Seed beds, they are used as seed beds due to excellent moisture holding capacity, and they are good soil binders as well. They are growing in dense mats that help to prevent soil erosion. Clear? These are the economic importance of bryophytes. Next, it is used as fodder. Medicinal uses from sphagnum. Sphagnol is the chemical constituent which is used to treat the skin diseases, and it has good antibiotic properties, and botanical research also it is used in spirocarpus, which is a liverwort. This is used to study the sex determination in plants. They are good pollution indicators and mineral indicators as well. Yeah, this is the table which gives you a clear picture on ecological and economic importance of bryophytes. You can take a screenshot of this and revise it, ma. Very important. This also, what all we have discussed, completely is tabulated. Fine, right. So, with this, complete bryophytes is done. Today's keynotes, we have discussed about the complete structure, description and life cycle of Anthoceros and Polytrichum. In gametophyte, both male and female sex organs, how they are formed, what is their anatomy, and how sporophyte is developed, what are post-fertilization events, and the life cycle, haplodiploidic life cycle is clearly being dealt. After which, ecological and economic importance of bryophytes with a clear tabulated form it is given out to you. Okay. So, this is the complete concept of bryophytes from Archegoniate. The next session on Archegoniate will be about pteridophytes, where we will be learning the general characteristic features of pteridophytes, and two type studies we are going to cover, that is Rhynia and Lycopodium. So, see you in the next class, and thank you.